Hey everyone, Mr. Murphy here. I'm going to be providing an introduction to the concepts of transcription and translation, which basically is an explanation of what DNA actually does. So remember that way back when we were talking about different organelles inside of the cell, we introduced that the ribosome was a very, very tiny little organelle that was responsible for building proteins. So really, just to get to the point, DNA is just the instructions for those ribosomes. And that's it. And I start with this because I think it's important for us to clear the air in terms of, we talk about DNA like it's this, super involved and complicated thing that's just running the entire cell. We use those words sometimes. We say that DNA controls what happens inside of the cell. And while I don't think that's really a lie, it really overshadows the fact that DNA has a very, very simple function of providing the code to make proteins. Those proteins then do everything inside of the cell. So the DNA itself isn't actually involved directly in all of these processes, but rather the DNA makes the proteins, or the DNA is the instructions for those proteins, and then those proteins actually do the job. So it's the ribosome that builds the proteins, and it's the DNA that contains all the instructions for those ribosomes. Now there's a few problems. The big one is that DNA is precious. Our cell only has one copy of DNA. And the reason why that's important is because if you want your DNA to be providing codes and instructions for a protein that maybe you need to make inside of that cell multiple times a day or multiple times a minute, I don't know. The problem with DNA is that you only have one copy. Imagine if you had a single sheet that was the instructions on how to do some important task. And, it's a task that you needed to be able to tell lots of people how to do. You wouldn't take that single copy and just hand it to a person and be like, hey, follow these instructions, and then, send me the instructions back because they're super important and I only have one copy. Right? The idea being that if you have something that is super, super precious and it's super, super important and you only have one copy, you don't really want to give that away. You might trust your friend to give it back to you, but if they don't, then you're really going to be out of luck. And that's the problem with DNA. It's too big, also. So because it's got that double helix structure and because it can be millions of nucleotides long, it's a very, very large molecule and very, very difficult, therefore, to get it out of the nucleus. Again, you don't want to do that anyway because it's so precious and fragile. So because of that, we need to find a way to take the piece of DNA that you actually want to give the instructions for, and we're going to make a copy of that DNA, and we're going to make a copy in the form of a different molecule, one that's smaller, one that's expendable, that we can, if you lost a copy of it, no big deal, we can just photocopy it again, so to speak. So that's what RNA is. So DNA stands for deoxyribonucleic acid, and that is the double helix structure that you see here on the left. RNA is the single strand copy of a segment of DNA. So let's say we had a gene that codes for amylase enzyme, which was one of the enzymes that's in our saliva. It helps to break down carbohydrates. Well, every time you make amylase, which is frequently, right, our salivary glands are constantly making enzymes because we eat throughout the day, and so we need a lot of amylase throughout the day. So our DNA has all of the genes for all the things that make up you and how your body functions, and so you only want that one little piece. And what happens is that that little piece gets turned into a strand of RNA, or gets copied into a strand of RNA. So the DNA itself is safe. It's like you took those important instructions and you photocopied them, and then you put those important instructions back away somewhere safe. But meanwhile, you've got this photocopy that now you can do things with. And that's what the RNA is. It's a copy of a segment of DNA. It's single-stranded, so it can leave the nucleus. And what happens is RNA has a couple special features about it, or not special, just things that make it different than DNA. First off, it's ribonucleic acid. It has a ribose sugar. It's single-stranded. That's important. That's a pretty big difference, and that's a big part of why it's easier to move around inside of the cell, because it's a smaller molecule. And then the other thing that makes it a little bit different is that it doesn't have thymine. It's the T, we usually have A, T, C, and G in DNA. The only thing that's different about RNA, it follows all the same rules, except anywhere where you would have thymine, it's replaced with uracil. And that comes down to the structural chemical differences between DNA and RNA. So it's not anything particularly special, it's just that thymine doesn't really work with the molecule, and uracil does. Chemistry reasons. We could go into that with more depth, when you take AP Bio or biochemistry or whatever. So this is a process that's called transcription. That process of taking DNA and making a copy of it in the form of RNA. So you would be taking just a single gene, just the one little segment of the DNA that actually codes for what you want, and you're going to make a copy of it in the form of RNA. It has all the same complementary rules. So basically you take your double-stranded DNA, you unwind it for a second, and then you take one of those strands and you start matching up. If there's an A on the DNA, then that's a bad example. If there's a C on the DNA, then you'd match it up with a G. If there's a T on the DNA, you'd match it up with an A. The only difference is that anywhere where you have an A on the DNA, that's going to match up to a U in RNA. So the same complementary rules apply. A's match up with U's, C's with G's, and you can reverse those. And what you end up with is a single-stranded complementary piece to whatever your DNA was. And that's what your RNA is. So it's kind of like the negative version of a photocopy. Okay? But it still has all that information that was on the DNA in the form now of RNA. And so I'm going to show you this quick little video clip. We'll see if this works. Okay, so I'll post a link for this so you can go back and actually listen to them talk about it. But what we're looking at here is a DNA molecule. You can see it. And this is a little protein here called RNA polymerase. And RNA polymerase is the protein that's actually responsible for turning the DNA into RNA. It's responsible for that process of transcription. Now, a few things come flying around here. These are different proteins that control the process. You don't always want to be turning DNA into RNA. You want to be able to control, so like when you need amylase, you can turn the DNA that codes for amylase into RNA. So watch as this starts, and you're going to see this little guy go zooming along the DNA. And this is an approximation of real time. It's very, very quick because it's so microscopic. And what you see is this yellow part that's flying out the top of this. And I like to think of it as like a piece of like receipt paper, that as this is running along, you've got this long receipt just kind of moving out of it, and that's what you see here. It's like a CVS receipt here. This is a giant thing that's just flying out. That's your RNA. So it's following along the DNA horizontally, and these yellow pieces are the different nucleotides that form the RNA. And that RNA, you can see, sort of grows and grows and grows and grows, and then when you get to the end of the gene, that RNA gets released, and at that point it can leave the nucleus, it can go to the ribosome and provide the instructions to the ribosome of what to build. So notice though that the DNA, as this is running along, it very briefly gets separated, gets unwound, but then it gets packaged back up immediately. So it's like you're taking that super important copy of a document, you might take it out of its little plastic protector, photocopy it, and then you put it back in the plastic protector again so it's nice and safe, but you've got that copy now to work with. Whoops. So here's an example. If we have this DNA molecule, CTAGAACGTATAAGC, and we're going to turn that into RNA, what will happen is this C will match up to a G, T will match up to an A would normally match up to T in DNA, but it will be a U because it's RNA. G becomes a C, A matches with with U, A with U, C matches with G, G matches with C, T matches with A, and so on. And what you can see is that we end up making this RNA molecule that is complementary to the DNA, following those rules of C's always match with G's, T's match with A's, A's match with U's, G's match with C's. And then you run along the entire length of the DNA. That's what transcription is. It's turning DNA into RNA. And I shouldn't say turning it, it's making a copy of DNA as RNA. The DNA itself is untouched in the end of this. The DNA stays the same, so that way we can come back and make another copy again later if we need to. Once that happens, so in this diagram you have a cell, and there's a nucleus here, it's the darker blue part. You have DNA, process of transcription, which makes RNA, and it's called mRNA here because it's a particular type of RNA that is called messenger RNA because it's going to deliver the message to the ribosome of what to build. Once that happens, that mRNA now can leave the nucleus. It's small enough, it's single-stranded, it can get out of the nucleus, and when it does that, it can get to the ribosome and provide the instructions to build the protein. And that process is called translation. So we transcribed it into RNA, and now we're going to translate that message and actually find meaning from it, right? Like the word translation means to take something and to put it into a different language or turn it into a different thing. So we're going to take this RNA and we're going to actually turn it into the building blocks of a protein. So translation is exactly that. It's the ribosome takes the code of RNA and uses it to build the protein. So based upon that code, the ribosome adds specific amino acids to a chain. So it'll take little sets of 3 RNA nucleotides. So 3 at a time, it'll take those and say, okay, well, it's these 3 letters, so that is this amino acid. And then it gets the next 3, and it says, okay, that's this next amino acid, and this next one, and this next one. Those sets of 3 are called codons, or sometimes called triplets. And so it's always reading by 3s, and for every 3 it turns that into an amino acid, and it keeps adding these chains of amino acids, one after the other, after the other, after the other. And eventually then, as that strand gets longer, it starts to fold in on itself because of different chemical reasons and chemistry and attractions and things, and that actually becomes the protein. So we went from DNA to RNA, that's transcription, and then taking that code of RNA and turning it into actual amino acids, that's translation. The process of translation follows really set rules, which is kind of cool and makes it pretty reliable. And what you're looking at here is a table that's showing you each possible set of 3 nucleotides and what amino acid they would code for. So there's 20 different amino acids that are possibilities for building different proteins. These amino acids, remember, an amino acid is the monomer or the building block of a protein. The way you read one of these charts is, let's do the first example, ACA. So let's say that the RNA has delivered to the ribosome a strand of RNA, and that RNA starts with ACA. When we look at this table, and I'm explaining this because you'll be asked to look at these tables and read from them, and at first it's a little confusing, but once you get it, it becomes very, very simple, the process. So on the left hand side you see UCAG, and it says that's the first base. So if we're going to do ACA, I find A, and here it is on the 3rd row. So right away I'm looking at the 3rd row. Okay. Then at the top it says now find the second base. So in our example I'm looking for A, C, A. So the first base was A, the second base is C, so I'm looking at the 3rd row, and I'm looking at the 2nd column because the second base is C. So I know I'm in this box here. And then the 3rd letter was A, so I can see ACA. And that tells me that this is the THR amino acid, which is something called threonine. So the exact names of these amino acids don't really matter. Just understand that they are different versions of amino acids, different shapes, sizes, chemical characteristics. Just they're very similar to like the idea of if you had a whole big pile of Legos, right? And you've got let's say 20 different shapes and sizes to those different Lego blocks, you could put them together in an infinite number of ways. You could make anything that you can imagine, assuming you have enough pieces. That's what's happening here. We have 20 different types of amino acids, and we can put them together in whatever order we want, as many of whichever ones we want, and that will be able to build this three-dimensional thing of a protein. And we can build pretty much any different shape, size, or thing of proteins as long as we have the code for it. Much like how in Legos, as long as you had the instructions and enough pieces, you could build whatever you want. You might be thinking, well, I don't even need to worry about these things on the side and the top. I can just look for the code. That's not wrong. You're not wrong. This type of chart does list out every single triplet or codon. It's not the most efficient way to do it, and some charts don't do that. So some charts would really rely upon you following the rules and finding the specific pattern of A, C, A, threonine. If we look at these other ones, GAU, so I go first base on the left-hand side here, I go down to G, and then I do second base is the third column, so I know I'm in this box bottom row and third column, and I find GAU, and it's ASP, which is aspartic acid. Again, doesn't matter the actual name of the amino acids. You'll never be asked to memorize those or anything like that. Just understand they are different building blocks of proteins. The third one, UUC. So I go U is the first row, U is the first column, and then there it is, UUC, and that's phenylalanine. Okay, different amino acid. Some of them, you'll notice, though, are a little different. So if I do UGA, so that's first row, U, fourth column, G, UGA, and it codes for something called stop. That's not an amino acid. That literally means stop. And so if you're wondering, how does the ribosome know when it's done building a protein? It knows when it gets to a stop codon. And so that's a segment of the RNA that's telling the ribosome, hey, you're done. Let it go. It's built. And that's what the stop codons do. We actually see a very similar thing. AUG over here is methionine, which is, in most cases, our starting codon. So that's telling the ribosome where the actual important part of the message begins. So there are different control mechanisms. There's a lot more detail to all of that we're not going to get into. So this is the whole process of transcription and translation. We started with DNA. We transcribed it, which means we turned that DNA into a copy of RNA. We sent that RNA out of the nucleus, and that RNA then gets to the ribosome, where the ribosome translates that RNA, or it takes the RNA and it turns it into a series of amino acids that are linked together, and that's a polypeptide, which can then fold up into our three-dimensional protein. I know that's a lot. We're going to take it in pieces and sort of work through step by step on this, but I wanted to give you this sort of somewhat quick overview of these processes, because it can sort of be overwhelming at first, but with some practice you'll see it's a pretty straightforward process of, sort of make a copy of the DNA and then turn that copy into a protein. And it follows really simple rules, so you just kind of got to practice it. Okay, so I hope that was helpful, and let me know if you have questions.